Let's start by saying, son, that evolution has been studied genetically using a very clever tool, molecular markers. But what is a molecular marker?
A molecular marker is a DNA sequence located in an easily identified region of a chromosome, and its heritage can be traced. A marker can be a gene or any sequence with unknown function. The markers that we use nowadays are nuclear DNA variations like single nucleotide polymorphisms, etc. On the other hand, the mitochondrial DNA contains the genetic code for 37 genes, and the ancestral markers used are single nucleotide polymorphisms that occur in the genetic code of the mitochondrial D-loop region. More than 50% of the world's population harbor H. pylori in their upper gastrointestinal tract, and it has been seen that depending on the geographical position, the strains differ from one to another, observing therefore different genotypes. Studying these bacteria in different populations, we can guess where they came from. With the Y chromosome as a marker, we can use different methods of substitution rates. One is obtained from human-chimpanzee comparisons, but the problem is that selective pressure in chimpanzee is higher than humans. Other is used to adjust the substitution rate to the rate of mutations of autosomal chromosomes, but the rounds of replication of Y chromosomes are more frequent. The ideal method is to collect and sequence some reliable deep-rooting pedigrees representing a broad spectrum of worldwide Y chromosome lineages. But why use mitochondrial DNA over the Y chromosome as a marker? We know that both of them are not mixed up by evolutionary mechanisms, but mitochondrial DNA has a larger number of polymorphisms and is passed to the maternal lineage, so that each individual has only one type of mitochondrial DNA. Also, human cells contain hundreds of copies of mitochondrial DNA genome, so in case the DNA is degraded or we have small quantities, it's easier to recover it. But how are these markers useful for the challenge of finding how populations have evolved? Information provided by the study of ancient DNA and actual DNA, jointly with findings in archaeological sites, etc., has led to a number of hypotheses on how the world's population today has become genetically as it is and how it has variated over time. The first theory says that those who first colonized a region are directly and solely related to subsequent generations in this place. Another one supports that advanced communities replaced other less developed. Theory 3 holds that the populations are established according to selective pressures in the area, but actually what has been discovered is that the Homo sapiens moved out of Africa to Mid-East, from Mid-East to Europe and Asia, and from Asia to Oceania and America. The theory of subsequent admixture says that some of these populations then moved into others and breeded back. They intermingled, even with different species that were already there for the first hominid migration. Let's take a closer look at some remarkable population movements that took place. In Asia, Homo sapiens breeded with Denisovans, giving rise to hybrids, which is the reason for having populations with a small percentage of pure Denisovan DNA. These populations divided in smaller groups that colonized Oceania and America through the Bering Strait. Neolithization. In Europe, there were 3 genetic events that determined the actual population's gene pool. The first one, the admixture between anatomically modern humans with Netherlands who lived in the same period. Scientists have discovered a relatively high percentage of Netherland genome in actual people. The last ice age. In this era, Europeans had to take refuge in 2 principal locations, the Mediterranean province and the Periglacial province. When temperatures became warmer, these groups recolonized Europe, but the Mediterranean province genome predominates nowadays. And the spread of Neolithic culture. Modern humans don't directly descend from pre-Neolithic populations that lived in Europe. Neolithic groups came from East regions and reintroduced their culture in Europe. So, human migrations as we know them are a really complex sequence of events, and this represents a truly difficult task for humanity to understand in order to give an answer to the question that has haunted us for so long. Who are we and where do we come from?